Okay, thank you so much for having me. In the memory literature, the hippocampus is synonymous with conscious, declarative, episodic memory. So, for example, if you're out at dinner last night, you can remember who you were with, maybe some of the topics of conversation, where you were in the world, what time of night it was. And most importantly, today you can mentally travel back in time to that event and relive it in a way. What's especially striking about episodic memory in the context of this conference is that we don't have this kind of memory for infancy later in life. And so what I'm going to talk about today is a bit of a story that's still being written that has kind of changed my thinking about episodic memory in infancy. Typically, people treat the lack of episodic memory from infancy as indicating that the hippocampus is immature and unable to support episodic memory function. And what I'm going to tell you about is a line of studies that suggests that the hippocampus is engaged in infancy and may be able to support the formation of episodic memories. Okay, so the story starts with, it's sort of a circuitous story, so bear with me, but the story starts with the finding across several of our studies and other people's work that the hippocampus supports more than episodic memory. So for example, we've done a lot of work on statistical learning, which is our ability to extract regularities across episodes. So yes, you might remember that particular dinner, but also you've learned more general things about restaurants, what they look like, the order of operations. You go into a restaurant, they greet you, you sit down, you order, you pay, you leave. Also spatial regularities, for example, in New York, what neighborhoods tend to have certain kinds of food or certain qualities of food. This kind of statistical learning we've shown in a series of studies in adults is also dependent on the hippocampus. Those studies involve, for example, showing that statistical learning activates the hippocampus in fMRI or an intracranial EEG, that the size of hippocampus and subregions of the hippocampus correlates with statistical learning abilities, that lesions to the hippocampus disrupt statistical learning, and so on. And so why I'm mentioning statistical learning in the context of infants is that infants are remarkably good statistical learners. The pioneering work of Jenny Safran, Dick Aslin, Alyssa Newport, Natasha Kirkham, Scott Johnson, many others have demonstrated that even very young infants, even newborns, are able to extract regularities in the auditory or visual modality. So we have a bit of a conundrum here. The canonical hippocampal function of episodic memory seems deficient in infants. In adults, we've shown that the hippocampus supports statistical learning, and infants are really good statistical learners. So it could be that hippocampus is immature and that infants learn statistically using other brain systems. That's one possibility. But another possibility that was pursued by a former graduate student, Anna Shapiro, who's now a professor at UPenn, her theory was that, in fact, the hippocampus supports both of these functions along different pathways. And so I'm not going to go through the circuitry of the hippocampus other than to say there are different routes through which information flows through the hippocampus. One of those routes, the trisynaptic pathway, seems to be supporting our ability to form discrete episodic memories for individual events. And another pathway, the monosynaptic pathway, seems to support our ability to extract patterns across events. So this was a computational modeling paper using a neural network that's built on the structure of the hippocampus to show these different functions of the hippocampus. From a developmental perspective, this is quite interesting because in tracing studies in non-human primates, these two pathways have different developmental trajectories. So this is in macaques, if you were to trace the connections in the hippocampus, you see the monosynaptic connection being established by juvenile monkey age, and you don't see the trisynaptic connection emerge until adulthood in macaques. So there's a differential developmental trajectory of these two pathways, which might help reconcile that initial sort of conundrum that perhaps the hippocampus supports statistical learning before it's able to store durable episodic memories. And so this licensed us to think about, well, is the infant hippocampus learning statistically? And to do that, we developed a series of methods. If you were to study the hippocampus in adults, you typically do this with functional magnetic resonance imaging. It's a deep brain structure away from the surface that's very hard to image with scalp EEG or with near-infrared spectroscopy. In adults, the methods are very clear, but in infants, doing fMRI research is very difficult, hopefully for some obvious reasons. For example, you can't instruct a baby not to move, and 1 millimeter of motion can really screw up fMRI data. So that's one example. Attention span, so putting a baby in an experiment, typically in adults, we get an hour and a half of data. And in babies, we'd be lucky if we got 3 to 5 minutes of data. So there's also a data quantity issue in addition to a data quality issue. In any event, I'm not going to go through the long story of the past decade of developing methods for doing awake infant fMRI. I'll put some links here. I'll bring this back up at the end if you want to look at it. Some of our papers and all of the data that we've collected over the last decade is all the published data is publicly available, as well as some software for analyzing it if you're interested. We've scanned more than 350 infant sessions. Again, this is a dense plot. The dark green in each column here refers to the amount of usable functional data that we acquired. And so you can see that on average, about 5 to 10 minutes of usable fMRI data, which is not a lot and requires us to be really creative about developing experimental designs that can accommodate so little data. If you're interested in learning more about infant neuroscience, Dick Aslan and I wrote a review paper last year reviewing both fMRI, but also other infant-friendly methods. Okay. I was tempted to summarize some of our results, but I'm not going to for the sake of time to focus on the question of episodic memory. But just to say that this is such a nascent field that we've been working with Rebecca Sachs's group, who is another leading group doing this kind of work, to try to find sort of general properties of doing these sorts of experiments. What are perhaps some best practices for making this work well? And so we have a preprint out now if you're interested in doing this kind of work. The earliest work in this line was conducted by a former graduate student, Cameron Ellis, who's a professor at Stanford now. I'm mentioning this because earlier Tim mentioned it. Our initial work in this domain was really in addressing questions about perception and attention. So Cameron did a study of retinotopic mapping. He also did a study on the role of frontoparietal control regions in sort of endogenous attentional control. And so this is showing a video of the task, the eye tracking that we perform. So there's a camera in the MRI machine, and then the brain images as they stream in. So I'm not going to go through the study other than to say it produces a really clear attentional behavior, which is looking time to targets as a function of attentional cueing, and also engages many of the same frontoparietal brain regions involved in controlling adult attention. Tristan Yates, who's in the room, she's blushing. I don't know why. This is beautiful work. Who's a former graduate student, now a postdoc with NIM Tottenham, has also conducted some really exciting studies in the space that may be relevant for thinking about infant consciousness. In particular, this is a study from a couple of years ago in which we presented infants with movies, and we used hidden Markov models to try to understand the structure of their experience. When adults perceive a continuous movie or stream, we don't actually experience it continuously. We experience it in sort of discrete events. And using data-driven methods, we're able to extract sort of what the event structure of an infant's brain is as they watch a movie. So that's sort of verging into saying something perhaps that might be informative for understanding the nature of infant experience. I'll skip over that for the sake of time. Okay, back to the main thread. So we had this hypothesis that the hippocampus might support statistical learning sort of earlier in child development than its ability to support episodic memory. So Cameron did a relatively simple study. This was modeled on work by Natasha Kirkham, in which we presented babies with a continuous sequence of fractal patterns. Embedded in that sequence were pairs. So after A, you would always see B next in the sequence. But after B, you could see the first of any of the other pairs. C would always be followed by D. E would always be followed by F. But the pairs would be randomly interspersed in the sequence, and there's no other cue to the boundaries between the pairs. So this is modeled very much on the sort of original Safran style syllable learning task. So we showed babies sequences that had a pair structure embedded in them, and we contrasted the evoked fMRI activity with sequences in which there were no regularities. So same number of items, same frequency of the items, but the transitions between fractals were not predictable. And so this is a very crude, kind of simple fMRI experiment where we're contrasting the overall level of fMRI activity for these two conditions. We're also going to look over time. This is a learning task. You don't know what's paired the first time you see this. You have to be exposed to the regularities. So the prediction is that we would see emerging differences between the structure and random condition. Okay, so this is just to give you a sense of what the task looks like. Babies are watching it. They're looming to keep them engaged. And then we're going to extract the pattern of fMRI activity for each of those sequences. We're interested in the hippocampus, and so we manually define a region of interest for the hippocampus based on anatomical scans. And we extract the fMRI activity from this. I'm just showing sort of the highlight result here, which is if you compare the activity evoked by structured versus random sequences in the first versus second half of the exposure, what you see is an emerging difference where by the second half of exposure, there's reliably greater hippocampal activity for the structure than the random blocks. This time by condition learning effect does not vary over infant age. You can tell I'm a bad developmentalist because we didn't actually pay much attention to age on the input. We're looking broadly across 3 to 24 months, but we use age as a variable that we try to relate to the effects. This is partly a practical necessity. It's extraordinarily difficult to recruit infants for this kind of research, so we take all comers. I'll skip over the rest of this just to say that this study provides evidence of that the hippocampus is involved in statistical learning in infants, perhaps as early as 3 months. Now, we didn't see an age effect on the statistical learning. This isn't to say we didn't see an age effect at all. In fact, if you look at the anatomy, there's really dramatic changes over this same age period. So this is showing the volume of the hippocampus from 3 or 4 months up through 24 months. So the volume of this brain region is doubling, but the learning related interaction did not change with age. This isn't normal. This is unnormalized, but it's the rate of growth of the hippocampus is faster than the global brain growth. Yeah. Okay, so this fits with that theory that that maybe the circuit develops faster for statistical learning than it does for episodic memory. But these results got me thinking, well, how good is the evidence that the hippocampus isn't encoding episodic memories in infants? Most of the work on infantile amnesia is sort of retrospective surveys where you ask older children or adults, what's your earliest memory? And people typically report 3, 4, 5 years old. There is some work in infants using tasks that have some characteristics of episodic memory tasks. The classic work by Roe v. Collier, we mentioned earlier, someone mentioned deferred imitation. And so there are some ways of studying memory, but trying to understand whether these are really the kinds of hippocampal encoding that's really critical for autobiographical memory was unclear. But we now had a way of getting at that. Using fMRI, we could image the hippocampus during an episodic memory task. So this is Tristan's work, which is going to come out shortly. And in this study, we basically replicated a design that was developed in the late 90s by Anthony Wagner and others called a subsequent memory design. John Gabrielli had a paper like this as well. So this is a study in which you present discrete items one at a time on the screen. In the adult version of this, you ask people at the end of the experiment, which pictures do you remember? Which pictures do you forget? Old, new judgments. And then you go back and you recode the fMRI data based on the subsequent behavioral responses. So if a person says old to a picture they saw before, that's a remembered trial. If they say new to a picture they saw before, that's a forgotten trial. And you can compare activity during encoding for images that were later remembered versus forgotten to index what's going on in the brain when you're forming a memory. We couldn't ask babies what they remember explicitly like that, so we used a preferential looking paradigm, a visual paired comparison, in which on the test trials we presented 2 items from the same category, one that had been seen before, one that was novel. After single shot exposure to a picture like this, babies tend to look more at familiar things. I don't want to make a global statement about that, but in general, we expected to get a familiarity preference because they just have this sort of minimal exposure to an image once before. So they're not habituated to the picture. In this case, we were predicting that the degree to which they looked at the familiar item would be a measure of the strength of memory that they had for that item. And so we're going to code the encoding trials based on subsequent familiarity preference. This is what a sort of baby doing this task looks like. Pacifiers are not good for head motion, but a necessity. And so you can see this is a test trial and they're looking back and forth. We manually code the amount of looking left versus right. This is very crude eye tracking. Here's a demo of what the task looks like. The crazy psychedelic background is to keep the babies engaged. These sort of discrete events are not baby friendly. Tristan could tell you about our first attempt at this where we did not have a background and the babies fussed out very quickly. And I think a test trial will come up here in a moment. Right, and so then we're measuring the, lateralized looking in that case. Okay, so again, same idea. We're going to extract a region of interest from the hippocampus anatomically, and we're going to compare hippocampal activity during encoding of the pictures where they later showed a familiarity preference versus trials where they didn't show a familiarity preference. And what we see across the whole infant sample is that greater hippocampal activity during encoding predicts later familiar looking. Interestingly, and I don't have time to go into this too much, but that circuit that I talked about, the trisynaptic pathway that supports episodic memory, is more based in the back of the hippocampus, the posterior hippocampus. And indeed, when we break down anterior versus posterior, these effects are really driven by the posterior hippocampus. Now, this is a within subject analysis where we're comparing encoding remembered versus forgotten or familiar versus unfamiliar items within participant. If you break it down and look across participants, these effects are driven by the babies that show the highest familiarity preference on average. And so this is breaking a median split on the strength of the familiarity effect. Now, whereas in the statistical learning case, we didn't see an age effect, here, based on some of the data that were talked about before, the behavioral studies of memory in infants, we hypothesized that there might be a transition around 12 months in the ability to encode these kinds of memories. And indeed, that although there was an effect in the overall sample, it was driven entirely by babies over 12 months. You can see 11 out of 13 babies over 12 months showed this effect, and there was no effect under that. This is also true in a more continuous type analysis. Final thing I'll show is that these effects are relatively specific to medial temporal lobe. So this is a whole brain analysis, again, showing that the greater activity during encoding of subsequently familiar items is really specific to the older infants. Okay, so those are the main data I wanted to show. Where we're going from this is to say, well, item recognition is a kind of episodic memory. But if you think about episodic memory more generally, it has many other sort of richer components. So, for example, it's not just about encoding discrete items, but also about the relationships between items, who, when, where, what, from dinner last night. And so a current student, Lily Bem, has been running studies inspired by work in adults by Charan Ranganath and Debbie Hanula, and some work by Jenny Richmond and Chuck Nelson in infants. A relational memory task where you see a scene, a face appears on top, and then at test, the scene appears again with two faces. And the extent to which the babies look more at the face that was previously paired with the scene indicates that they formed an association between the face and the scene. And we're looking at what happens in the hippocampus during encoding of pairs where you later show this associative memory. She's also interested in looking at spatiotemporal relationships. So in this case, you learn a sequence of these sort of fun cartoony characters that appear at different locations. And we can test memory by looking at predictive looking. So if you remember a sequence and you see 1, 2, 3, do you look to the expected location of the upcoming item? Okay, the final thing I'll say, and this is very much still ongoing. We just finished a longitudinal study to try to look at episodic memory in infancy over the kinds of time scales that we're usually thinking about in the context of infantile amnesia, thinking back as an adult to being a baby. Over the span of months or years. We've collected a longitudinal data set where we followed 7 families that we had them record home videos across infancy, and we scanned the babies every 3 months to sort of track how their hippocampus responds to videos that were recorded from their own experience versus from another baby's experience. Sadly, I don't have any data to show you yet on that, but what I will show you is that we ran a pilot study, which is, there's a great data set called SACAM, Mike Frank and others collected, in which they put a head-mounted camera on 3 infants and tracked them for years. This is an example of the kind of video that was recorded.
Okay, are you ready? Let's put the stick at the bottom because we could hurt ourselves if we carry it while we're doing the slide.
Right.
You made it, we made it.
I like it. No.
What do you want? Oh, you want your stick?
So these videos were collected by somebody else, but we brought 2 of the 3 families to New Haven and we scanned the kids when they were older. But we had their experiences from when they were babies. The videos were collected sequentially. So the latest kid who did this was 3 years old when we scanned them. The first kid was 9 years old. And so this sort of pseudo, case study, pseudo longitudinal thing where we can compare a 3-year-old's response to watching their own versus other experiences and a 9-year-old's response watching their own versus other infant experiences. And just to jump to the key result, only the 3-year-old's hippocampus distinguished self versus other, not the 9-year-olds. So this indicates that not only are infant experiences encoded, as we had shown before, but also that they might persist into early childhood. And what we're working on now is trying to figure out, why do they become inaccessible? And I have ideas about that. I'm happy to answer in the question period, and I'll just leave up the summary and take any questions. But thank you to everybody.
Mentioned that item recognition was an example of episodic memory. Couldn't it just be familiarity without knowing why something was familiar?
Yeah, so in the episodic memory literature, there's under the umbrella of episodic memory, people distinguish between familiarity and recollection as 2 forms of remembering. And so we do think that it is familiarity with the stimulus that's driving looking. But even in that case, it's one-shot encoding. So you've seen this picture just one time before. And so it's not like a priming type of familiarity where it's this long-term habituation or where you're seeing something back to back. Here, there's many intervening items and there's a lag between the initial presentation and the repetition. So we think this kind of familiarity is more hippocampally dependent. But yeah, we want to look more broadly at things like recollection. Can they take a partial cue and sort of fill in missing details?
Yeah, so just building off of that point, it's not, I'm not sure how you'd be able to, so like my understanding of episodic memory is like you're saying recollection, this memory of being there, of having a sense that you saw this before. And so with the faces, yeah, it might just be a matter of familiarity. And so you reply to that, but then I'm curious about this other study that you had to like go over to see if this might help with the potential episodic memory of babies. And you were talking about the films,
Yeah.
and you were talking about how we see that adult brains watch films through events.
Yeah.
But you didn't describe whether or not what the research showed about babies watching the films, if they're seeing just a constant stream or through events. So I was wondering if you could talk about that research.
So we haven't linked that study to memory, but I can tell you the result of that study was that infants do show systematic structured events while watching movies. They don't perceive it continuously, or their brain regions don't, but they have fewer events. They actually have longer timescales of processing in most brain regions than adults. Adults carve up the movie into more events than the infants do. And it's not just that the infants have a blurrier or noisier representation. We think they have a sort of a clear representation, but the window of integration over which the brain activity is stable is longer. How this relates to memory is a really interesting question. This was Tristan's study, not my study, but yeah. I was an author, but she was the one who did it. Yeah. Yes.
Thank you. I'm following up on that lovely studies.
Thank you.
So on the study you ran with the slide,
Yeah.
I mean, there are old data showing that eventually you remember yourself from the outside.
Yes.
You don't remember your father's feet.
Egocentric.
But if it was a father, but even on the earlier study where you had the faces embedded in the scenes,
Yes.
I wonder what would happen if those were little vignettes.
Yes. I love the question. Yeah, what you're referring to is that, over time, memories get somewhat decontextualized and you represent them maybe more allocentrically or even ultracentrically from somebody else's perspective. We don't know here how like the frame of reference changes. What I showed is that the 3-year-old's brain does respond more to watching their own perspective than watching a video with similar content that was recorded from another baby. But what we couldn't do, because the videos had already been collected, is manipulate sort of what perspective they're viewing their own event from. But that would be a really interesting study. And the hippocampus in the domain of spatial navigation is really important for forming allocentric representations of the environment. And so linking those functions would be really cool. Yeah.
But is the second question about giving them vignettes where the people and the scenes make sense?
Yeah, yeah. So you mean actually, how would you give it to a baby, a vignette? You'd show them a movie of
Right.
something
a short
happening?
vignette,
Yeah.
someone taking something,
Yeah, yeah,
moving,
yeah.
interacting
And then change
in
the
some
perspective.
way
Yeah.
that would involve presumably the hippocampus integrating
Yeah.
those different aspects
Yeah,
of
we haven't done that. It's a great idea. Yeah, thank you.
I have a quick question.
So a dominant theory of infantile amnesia is that the categories in terms of which you see the world change with development.
Indeed.
And that's the way that the child encoded the information. It's a version of Barbara's.
Yeah.
But you said you have some ideas. What?
Yeah, thank you for the prompt. There's some of my favorite research in this domain has come from rodents. There's a couple of really elegant mouse studies where you have a baby mouse learn something. Could be fear conditioning, but it could also be a spatial learning type task. And then if you let them grow up, 20 weeks later, and you put them back in that same context, they don't freeze. They don't have the fear memory. They don't go back to the reward location. So it's a model of infantile amnesia that people have used. But the more recent papers, what they've done is use optogenetics to stimulate cells in the hippocampus during the baby learning experience. So they're tagging some neurons as being part of the memory. And then in adulthood, you put the mice back in the familiar context, they don't show the learned behavior. But if you stimulate the hippocampal engram, then they express the behavior. They freeze or they go back to the reward location, which says that the hippocampal memory is there. But just being in the environment, the sensory cues of the environment aren't sufficient to reactivate it. And I think what you're getting at is the reason why. It's not that the hippocampus is immature in infancy, it's that the cortical sort of mantle through which our experiences are represented that provide the input to the hippocampus are changing dramatically. And I think this subsumes various theories related to language, related to categories. So what we're working on now is developing ideas about how to measure that. There might be some cortical domains that are fairly early developing or consistent where you might actually be able to get back to the hippocampal memory, maybe olfaction or maybe face perception. There are other domains like language or orthography or categorization where it changes dramatically. So you can look at the similarity of encoding retrieval kind of representations to try to predict whether you can get back to an early experience. But that's the ongoing idea. Yeah.
Fascinating findings. I was wondering if you had further thoughts. You mentioned navigation.
Yeah.
It's like I've done some work with roboticists where they use the hippocampal system basically to inspire architectures for the SLAM problem, or simultaneous localization and mapping. And like there are models of, for instance, of much of high level cognition, including like ways of like dealing with counterfactuals, referencing some of like Hassabis's old work and in terms of like generalized navigation.
Yeah.
And I guess I was wondering like what you thought of that framing. Is it like competing, compatible? And one more thing is if you had thoughts potentially on like human uniqueness of the hippocampus. Like there's old debates about like the hippocampus minor, they're kind of discarded, but then more recent things about like maybe there's less pattern separation in the human hippocampus, and ways in which that might like in ways human cognition might be distinct.
Yeah, no, I think it's the latter question's quite interesting. My current view on that is that maybe it's not so unique, not as unique as sometimes people think. We find pattern separation in adults and other things. I think what's perhaps unique to humans is the capacity of our memory, the fact that it's not always spatial. There's some sort of species-specific things in other animals that don't necessarily apply to humans, but the basic circuitry and some of the basic computational principles are somewhat conserved. With respect to the first question, I think it's a really interesting idea, to think about, to the extent the hippocampal system is available in infancy, what other aspects of cognition is it supporting? It's been implicated in many other aspects of cognition, and those haven't been considered so much in the infant cognition. That hasn't been the motivation for thinking about those functions in infants, but that could be a quite interesting sort of framework. There's a lot of work, for example, showing the hippocampus contributes to value-based decision-making, to attention, memory-guided attention, and so on. And so thinking about some of the canonical infant cognition work by Susan, by many other people through the lens of hippocampal memory and hippocampal systems could be really interesting.
Yeah, so I was wondering what would be the relation between those experiences that are encoded and consciousness.
Yes.
Are you
I
assuming
was trying to
that
avoid that question.
are you assuming that they are that it requires consciousness to encode those experiences, or are you approaching experience or with conscious experience, or we might have cases where we're encoding those representations without consciousness?
Yeah. So, I was really inspired by Tim's talk earlier. And, I think to some memory researchers, hippocampal memory or memory of one-shot experiences like what we showed could be thought of as a marker of something that tends to be conscious in adults. From a memory researcher perspective, I've never put as much weight on consciousness as sort of the defining characteristic of episodic memory or hippocampal processing. Folks like Larry Squire and others have really made that the centerpiece of what the hippocampus does. I have less of a stake in that. I think most episodic memories are formed incidentally, right? We're not sitting there at the dinner saying, I really need to remember this. But the actual sort of process of remembering is often a very conscious experience, the sort of Proustian, zooming back to the past. And so understanding the nature of that process, the sort of recollection, the reinstatement, recall, I think that would maybe speak to this more specifically, but we haven't done those kinds of experiments yet.
One last question, Dave.
It's just on this point, you mentioned John Gabrielli, and I remember back in the 1990s, he used to give talks with titles like Consciousness as the Gateway to Memory and Attention as the Gateway to Memory, kind of implying that without consciousness, you don't get the kind of serious encoding and memory. I'm just wondering, what is the status of that hypothesis 30 years on?
Okay. Well, I think attention certainly, for the reasons we were talking about before, modulates cortical representations in a way that influences what gets input to the hippocampus. So there is attentional modulation of both item and relational memory for sure. Whether you need to be conscious, how we define that, I'm not sure. Right. In order to encode a memory is an interesting idea. I don't think it needs to be intentional. It doesn't have to be your goal to encode, but it is the sensory experience that is what is being stored in memory. It'd be interesting to try to decouple that. I don't know. But,
thanks.
Yeah.
Okay, thank you very much.
Thank you.
Okay, so I will tell you today about some work that's part of a longitudinal study that has been conducted in Copenhagen, which is led by Katharina Begusch. And in this study, we were actually not primarily interested in consciousness or metacognition, but rather in curiosity, in the sense of information seeking. So maybe rather this type of inner drive that we heard a little bit about yesterday. But obviously, information seeking has a lot to do as well with metacognition, which we can define as the ability to evaluate one's own informational states and the efficiency of one's own learning in order to regulate subsequent cognitive activities and behaviors. So infants historically have been denied that capacity because it's been thought to require some maybe conceptual or self-reflective capacities that non-verbal populations such as infants or animals are thought not to be able to use. But we have some evidence that there might actually be some at least rudimentary form of metacognition in infants. And so you've heard about the results from Louise Goupil and Sid Quitter's study yesterday. So they've shown that infants are able to ask for help and to persist in their decisions in a way that is consistent with them tracking their own states of uncertainty. They've also shown that infants have a neural signature of internal monitoring of their own errors. So that's the error-related negativity, or the ERN, which is a negative deflection that appears in frontocentral electrodes after one's made an incorrect as opposed to a correct decision. And so very briefly, you've heard about this yesterday. In their paradigm, what they're doing with these infants is that they're cueing a side of the screen with a face that's briefly presented, and then the infants can make a saccade to the cued side to then see the appearance of a reward on the same side. So they have to actively make a saccade themselves in order to make a decision in that task. But they're still subjected to the sequence of the task itself. So they're not controlling that. And in our experiment, what we wanted to do is to try and replicate these findings, but also look at some further questions that remain unanswered, such as whether these rudimentary metacognitive capacities are in fact utilized by the infants, and whether it might guide their behavior in contexts of free information search and learning. So while both metacognitive monitoring and metacognitive control could work in a non-reflective level, to develop a more explicit and a more reflective form of metacognition. Probably this is quite tightly linked also with the development of self-awareness. And so that's also something that we wanted to look at, and to kind of look at the potential link between these different abilities. And by studying infants, we have the unique opportunity actually to investigate the developmental changes in possibly the only population that goes from non-verbal, pre-verbal, and pre-conceptual metacognitive capacities to more reflective abilities later on. So here, by following the same babies over time, we can actually capture these developmental changes and then look at whether the early metacognitive capacities might contribute to the emergence of self-awareness. Great. So how we approach this is through a longitudinal project. So we've been following 124 infants that we've seen at 12, 18, and 24 months. We've just finished the data collection for this in January, so we have not looked at all the data that we have collected. And so today I'll focus on the data from the 12-month-olds mainly, but I'll bring in some components with the 18-month-olds. And so what we do at 12 months is that we use a gaze-responsive paradigm with eye tracking and concurrent EEG recordings. And then at 18 months, the same infants come back and we do a mirror test with them to test their self-recognition. So probably you know how this works, but very briefly, what happens is you put a mark on the infant's nose without them being aware of that, and then you put them in front of a mirror, and you try and see if by seeing themselves in the mirror, they make the mapping between what they see in the mirror and their own body, and reach directly for their nose when they see this unexpected mark, which gives us evidence that then they have this sense of self. So 18 months is conveniently the age when about 50% of Western babies are passing this test. So we can try and compare those that do to those that don't, and then link all of this back with what happens at 12 months. Great. So in practice, how we do this is that we use a match to sample task at 12 months. So the infants are taught that finding a matching card is rewarding. And we use gaze contingency for this so that we can look at free and active information sampling, because the infants are controlling what's in the screen themselves. So this is what it looks like. It's loosely based on the game memory. So they have to make pairs of identical objects. But here we only have 3 cards. And first, they see two of the cards that are visible with objects depicted on them, and then a top card that is face down. And they can visually explore those two bottom cards for up to 20 seconds. And the moment they look at the top card, then this card will highlight, and then if they keep looking, it will flip. And that will flip the entire set of cards so that the same object as either one of the two cards they saw before is now depicted on the top card. And at this point, they can look back at either of the bottom options to try and make a pair. So if they look at a card, it will highlight and then eventually flip. If that was the match, then they also get a reward. If they do not find a match, then the cards are just silently fading away. So we don't do some negative reinforcement here. So this is what it looks like for an actual infant. So this is the infant gaze as a green dot, which hopefully you can follow. And so they're first just triggering the start of the trial. And then they're in the exploration phase now, so they can look at the cards freely. They just looked away. Whenever they look back, they're back now. Now they're in the decision phase, and they picked an option. That was the correct option. Yeah, okay, okay, so they get a reward. And so here they found the match and made a correct decision. So another feature of our design is that we're using an adaptive difficulty throughout. So we can make the match more or less difficult to find by making the images more or less similar that we present them with. So they can have very different images like an elephant and a strawberry, but then in the harder level of difficulty, they could have items from the same category, which here is vehicles. Next level, they would get the same subcategory, which is here cats within the animal category. And then in the hardest level, they're even of the same color, which really makes it quite challenging for them to find a match. And they don't go through these levels randomly. They actually go through them in a way that depends on their performance. So they start off at the easiest level. Here you can see correct trials in green, incorrect trials in red. And so as soon as they make two correct answers in a row, then they move up difficulty level. And again until they make two incorrect answers in a row, and then that moves them back down. So that ensures that we can have a good proportion of both correct and incorrect trials, and then try and compare them in our analysis. Great, so we have a set of hypotheses of what we think might happen in this context. We're interested in metacognitive monitoring, and if infants indeed have this capacity, we think that they should be able to detect when they're making an error, and that this should be reflected in their brain activity. So we can look both at this internal monitoring window right after they made a decision, which is the ERN window, but we can also look at the feedback window and look at the FRN signal. To look at metacognitive control, we use eye tracking, and we think that if infants are able to use some sort of metacognitive control, they should be able to adapt their gaze behavior with respect to their prior mistakes. And then the last thing we look at is the emergence of self-awareness in relationship with these abilities. And we think that the early metacognitive monitoring and control abilities might link with the later emergence of self-awareness. Great, so we'll start with the results in terms of metacognitive monitoring. And so the question here is, do infants detect their own errors? We look at their EEG activity to look at this. And we've based our analysis on the study from Luis and Sid. So we're using the same set of electrodes that they used. There's 58 infants out of the 124 that were tested that are included in this analysis. And they have to have at least 6 trials per condition to be included. So here we're looking at the kind of decision phase of the trial when they have explored the bottom 2 images here. They flip the top card, and then they have to make a decision. So we align the signal with the moment that their gaze enters one of the 2 cards before it highlights or flips, just when they make the saccade. We baseline with a previous window, and then we can look at the correct answers in blue and the incorrect in red. And so if we do a time series analysis of these two signals, we find two significant windows when the correct and incorrect brain activity differs in these kids. And that's a little bit different, but I think still quite easily conciled with the results from Louise and Sid. So in their study, they looked at a later window, which aligns quite well with the one that we find, but they did not look at an earlier time. If we look at the shape of the ERN, though, I think they might have an earlier peak, similarly to the one we find. And actually, initially, they found a really long time window in their time series analysis that was composed, that included both our early and our later peak. And then they focused on the peak that was the most negative. So I think even though there's a few differences here, probably we're kind of agreeing quite well in what we find in these infants. These responses are also in areas that are well centered on the electrodes that they found. So all in all, I think we replicate this finding pretty well. Then we can also look at the time of the feedback. So the infants have highlighted the card and now it's flipped. So they get a visual feedback on whether they were correct or not. And here again, we find a significant window when those two brain responses significantly differ, which happens in the same areas than the ERN signal, which again goes well with the literature on the topic. Great. So I think with these results, we're validating our first hypothesis that infants at 12 months probably have some form of metacognitive monitoring of their own errors. So we can look now at the metacognitive control part. And so here, the question is, do infants adapt their behavior following an error? And for this, we look at trial T, in which they've just made a choice, and that choice could be either correct or incorrect. So we just keep notes of the accuracy of that decision. And then we look at the next trial and what kind of behavior they have in there. And we try and see if there might be a difference between the trials when they've just made a mistake and the trials when they've just been successful. Here, we're actually discarding all the trials when there's a difference in difficulty between trial T and trial T+1, so that we don't introduce a sort of confound in how difficult it is. Great, so we can start by looking at the exploration phase, actually, when they just visually look at the cards they're presented with. And here we have several measures of how they're exploring. So just the time that it takes them until they trigger the next phase, the number of fixations that they make, and the number of switches that they make between the two visible cards. And whatever measure we look at here, we do not seem to find significant evidence that they are adapting their exploration to their previous mistakes or successes. But we can also look at the next phase, which is when they have to make their decision. And then here we can again look at how much time it takes them to make their decision. And there does seem to be a difference between whether they were just correct or incorrect, in the sense that they take longer after making a mistake. We also look at the proportion of trials when they just do not want to make a decision. So after 10 seconds, without looking at either of the bottom cards, the trials just automatically stop. And here there does not seem to be a significant difference in how they do that, depending on whether they were just correct or incorrect. But they do take longer to make their decisions after a mistake, which I think is a way to verify also our second hypothesis that they have some form of metacognitive control in this setup. So we can look now at the last part of our question, so the relationship between these metacognitive abilities and self. And so we can start with the monitoring part, and so go back to the brain activity of the infants, both in the ERN and in the FRN window, and look at the scores in the mirror test that they had 6 months later, and then go back to their brain activity and see if there was anything different going on 6 months earlier. So if we look at these two ERN and FRN signals for the windows that we identified earlier, we see that in the group that does recognize themselves at 18 months, there is a significant difference between the correct and incorrect brain responses, both in the ERN and the FRN part of the signal. But for the group that does not recognize themselves, there is no evidence that the correct and incorrect signals are different in the ERN window. There might be something in the feedback window. Importantly, those are kids that do not show any difference in their general development that we looked at through a questionnaire that the parents filled in. So it seems like it's much more of a specific type of ability that we're looking at here, which is more related to error monitoring and self than just fast development and performance overall. Great, so now we can look at the relationship between the development of self and the metacognitive control. And again, we split the infants based on their 18 months score in the mirror test. And then we look at their behavioral adaptation to their errors in the exploration phase first. So again, we have different measures here. And pretty much for all of our measures, what we see is that there is a significant difference in the difference between prior correct and prior incorrect between the two groups in the direction of the recognizers taking longer to explore after a mistake. If we look now at the decision phase, we do not see a difference between the two groups, either in terms of how long they take to answer or in terms of how much they opt out and just skip the decision between the two groups, which I think is interesting because the first variable, how much time they took to decide, is the one that we found in the whole group initially. So it seems that this one is not specific to the kind of mirror label of the infants, but more the maybe more instrumental phase of the exploration that comes before the decision. Great, this was from before. So now, hopefully with these results, we can agree that we have verified some sort of link indeed between the emergence of self-awareness and earlier metacognitive abilities, both in terms of monitoring and control. And so to summarize a little bit what I've shown you with all these results, what we found is that there is some evidence for metacognitive monitoring in 12-month-olds that we see in their ERN activity. There's also evidence of metacognitive control. They seem to adapt their decision-making following a mistake. And then the link with the emergence of self-awareness also seems to be present. So infants that show evidence of self-awareness, self-recognition at 18 months also show evidence of internal metacognitive monitoring and control at 12 months. And we did not see the same relationship in the infants that did not recognize themselves at 18 months. Great, so to conclude, already at 12 months, it seems that infants do demonstrate rudimentary forms of metacognition, both in terms of detecting their own errors, so metacognitive monitoring, but also in terms of adjusting their behavior, so metacognitive control, in a free infant-led paradigm. The other thing we find is that early metacognitive capacities seem to be related to the emergence of self-awareness. And so that raises the idea that maybe these functions develop in concert. That's an idea that has been proposed by other people before. You can ask me about this if you want. But yeah, there might be quite a tight link between these two. So thank you so much for listening to me. I would like to thank my PI, Katarina Begush, our technical team, army of students that have been helping us, and obviously all the families that joined the study. Thanks.
So this is very interesting. For some of the box plots that you showed with significant differences, just eyeballing it from my seat, it looks like the results are being driven by a subset of infants. There are a lot of bars that are just flat, and then you've got a few that jump way up. Is there some way that you could tease those apart? Is there a special super performing group of babies that is extra sparkly or something?
Yeah, you're entirely right. I think there's a lot of variability in there, but both in the infants and in the trials. So we're also trying to maybe keep more of the data of what happens throughout the experiment to maybe try and look at more kind of fine-grained differences from one trial to the next, for example, depending on the value from before and things like that. It's a bit difficult because we're quite exploratory here, so we have a lot of variables. Those are not corrected. The EG was corrected for multiple comparisons, but those are not, because we're still trying to figure out exactly how to include all of these different things we want to look at. We're also actually interested in how infants adapt to perhaps errors that they anticipate. So to the difficulty of the trial, to how error-prone the trial might be. And they also seem to adapt to the difficulty level. But then if we include all of this together, it's a little bit too many things at the same time. So we're actually in touch with a statistician to try and find a good way to look at all of that together. So this is still kind of work in progress.
Yeah, this is very nice. First of all, glad that you can actually replicate quite well this early study. And you have two questions. So the first one was about the first component and what do you make of it? I mean, it looks very similar to the earlier studies, but my most important question, sorry, is like, why would we expect actually such a strong correlation between something which is about bodily awareness and something which is about basically like decision making? Of course, those are two internal mechanisms. They're both linked to roughly to self-awareness, but those are still very, very different components. But it's really nice that you find this difference. But I'm just like still trying to understand whether there's a model mechanism which is driving both of them, or whether they're just like they're just correlated because they maturate at the same time.
Yeah. Maybe I'll start with that one. So there's actually some work in the curiosity literature that has had this idea that they might be related. There's been no evidence for that. It was just a bit of a blue sky idea. So that's work from Pierre Voudeyer and Frédéric Kaplan. And the proposal that they have is that when infants are starting off, they maybe don't have this sense of self as we adults have it. So they receive tons of inputs from internal input, external input, and that's just more or less information to learn from without distinction, really, possibly. While they try and predict what happens and make sense of all that information, this information that is actually coming from themselves might be slightly easier to predict, or at least cluster together with different inputs. And so there might be this sort of fuzzy concept of self that emerges from the necessity to make sense of all that information. And so that's how then those early abilities might be linked. And then as they complexify, maybe the link also becomes less important. Hopefully that answers your question. And then in terms of the ERN peaks, so I think the early peak is quite in line with the timings that they find in adults. It's a bit later, but that's what you would expect in infants. It's the same type of topography. So I think probably that is quite similar to what they describe when they look at the ERN in adults. And then they have a later peak around 400, which actually is positive. We have a later peak at the same time, which first of all probably isn't the same thing in infants, which is negative now. I am really not sure what that would mean. In your study, actually, I think I've cut it, but yeah, you find this later peak even later. Because at 500 milliseconds, we have the feedback. We do not have a chance to look at that. But I'm not exactly sure what would happen because there's a big time lag and those are much more integrative processes. So I don't know. I think there might be quite similar, the early and the late peak, but I wouldn't know what's the distinction.
I'm a little bit confused about exactly what happened here. And what I'm wondering, so you have when the card was fixated, but when is the item revealed? Is it right then?
No. So this is internal monitoring. So they
it isn't revealed at all.
It is revealed afterwards. Sorry, I didn't give you the timings, but basically, so when they look at the top card,
first it highlights after 200 milliseconds of looking at the card, and then for an extra 300 milliseconds of still looking at the card, then it flips. We allow for the infants to look away a little bit in that time, so there's a tiny variability in how much time it actually takes from the fixation to the flip, which is why we entirely realigned the FRN signals to the actual flip of the card, but more or less it's 500 milliseconds between the two.
So could you go back to the previous slide, the one that you were on before? So I see, so that's long after the graph that you're showing here. So is what they're, okay, so I'm just wondering what the form of the metacognitive awareness is taking in the kid's mind. Is it, there is a bicycle, so look at the upper left, there's a bicycle and I have an image of a bicycle there, but I'm being told that that's wrong? Is it, I mean, what is the error? What?
They're not.
They haven't gotten error.
It's just visual feedback. So they're still evaluating based on the fact that these two images are the same or not, whether they think it's correct. There's no reward or anything.
Is this like a matching, failure to match? But what is the failure to match between?
So I mean, so when maybe that will like going from another angle, maybe that will feed into your question. We've had some worries, for example, about the extra hard trials in whether they will even understand what was an error or something, because the images are so similar by then. So maybe these have to be treated slightly differently. But if you have an elephant and a strawberry here, I think for a 12-month-old infant, it's quite evident that they're not matching. And they've been taught, there's a whole familiarization phase that goes before that teaches them that they can flip cards with their eyes and that they should match them. In the familiarization, they actually get 2 chances to match the card, and all the cards are revealed once they pick an option. So they are given a little bit more of a, we're taking them a bit more by the hand to try and teach them what's happening. There's no guidelines, it's all implicit, but we think that they understand what they're trying to do, for some of them at least.
Okay, so then in that case, maybe it's a internal sign of uncertainty.
Yeah, I do not argue necessarily that this is not related to uncertainty. I think correct, incorrect is a very kind of experimental binary setup that probably has more to do with uncertainty.
But you know, the term metacognitive monitoring suggests that there is another state that is trained on the first order state and that is saying something like mistake, truth. But I'm beginning to wonder what the rationale for that is. Maybe, I mean, suppose it's just a sign of internal uncertainty.
Well, both of those are matter. Wait, this is the last couple we've gone over.
This is only about it. Here there's 2 things happening. First, the decision whether to go right or left. Yeah. And then there's an evaluation of whether this was a decision, the right decision or the wrong decision. So there is a second order aspect.
But it could be that, or it could be a reflection of your own uncertainty. You can't really decide between the two.
No, because if there was a reflection of uncertainty, you would find it all as well in the incorrect side, when they go to the incorrect side. It's not just uncertainty. It's, that's why we call it metacognition.
That's right. Yeah, you're right. You're right.
Yes. Correct.
Cool.
Or incorrect.
I just want to add something about this very quickly. I think one point that makes it important, whether it's uncertainty or not, is that there's also the metacognitive control part. So it does have an effect on behavior, whether they've made a mistake or not. So we can discuss whether that's really metacognition or not. But I think because it affects behavior, there's, I would call it at least a rudimentary form of that. And then the other thing we can look at, which is a bit more just eyeballing and qualitative, but if we compare the recognizers and non-recognizers, there's some quite interesting levels of activity here. So in the recognizers, the correct answers are really at 0. I mean, this is obviously baseline, but it doesn't really change from the baseline. The incorrect is quite negative, but in the non-recognizer, the group of non-recognizers, the correct and incorrect are at the same level in the negative side of the signal. So it might be that they don't know whether it's correct or incorrect, but they know that they don't really know that, which is then, I would call that uncertainty. I'm not sure, I really wouldn't want to make a distinction that strong between correct, incorrect, and uncertainty.
All right. So I'm coming to you guys from the University of Vermont and the Complex Systems Center. And I want to preface this by saying there is a sort of minimal amount of mathematics that I had to include in this. I've tried to make it as painless as possible, but you are welcome to ask questions if you're not following along. This was also a collaboration between my people and the people in Finland, and the people in Finland were the baby people. So I'm happy to answer lots of questions about mathematics. I have less capacity to answer questions about baby development because they're on the other side of the Atlantic Ocean. So we have been talking a lot over the last 2 days about consciousness as this sort of thorny philosophical problem. And as I was making these slides, I was thinking, you know, why just stop at one thorny philosophical problem when you can have two for twice the price? So I want to talk to you guys about emergence, which is another one of these perennial sort of bugbears. And hopefully it is not controversial to say that consciousness appears to be an emergent property of brain activity. I realize that there are some philosophers who may disagree. Hold your tomatoes until the end. And I'm just going to ask that you give me this one for the purposes of like the rest of the talk making sense. And I think the good evidence that consciousness is an emergent property is that we can perturb it. You know, you can give somebody LSD, you can give them propofol, you can hit them on the head with a baseball bat. And by changing the structure of connections between the brain and the dynamics of neuron-to-neuron communication, the quality of consciousness changes. So there is some sense in which, you know, what we experience phenomenologically is a function of the collective gestalt state of the brain. And so I would propose, and again, please just give this to me for the time being, that if we could create a formal algebra that could quantify the degree to which a system is capable of displaying any kind of emergence, we could then kind of backdoor our way into a measure of whether that system might be able to support consciousness specifically, assuming that consciousness is just a specific kind of this more general property of the general phenomena of emergent properties. Okay. And so this comes from a sort of a longer lineage of work attempting to find quantitative measures for consciousness. People talk about a thermometer for consciousness. I don't like that. I like this term, a lethiometer, and 10 points to whoever knows what the root for that is. And actually, some of this and so, you know, there's been a lot of work, you know, looking at things like Lempel-Ziv complexity, fractal dimension, sample entropies, tons of entropies. And Lorena talked a little bit about some of this yesterday with her work. I find a lot of these measures of complexity to be quite unsatisfying, typically because if they are entropy-based, they will be maximized in the case of random noise, you know, and it seems very odd to me that if you were to take a brain and put it in a blender, that, you know, your brain smoothie would, yeah, technically be more complex than a brain, but it's probably not any more conscious than the brain is. And so it would be nice to have a measure that really captures this idea of emergence, you know, that there is something happening in the whole that is not trivially reducible to its parts. And so now we're going to have to do a quick foray into the wonderful world of multivariate information theory, which is this formal logic for describing how parts of a system will disclose information about each other and the whole system of which they are a part. And there are two particular sort of flavors of interaction that I want to highlight here, okay? The first is redundancy. And this is sort of associated with synchrony. This is information that you could learn about anything that you could get by observing X by itself, or Y by itself, or Z by itself, right? If you think about a synchronized time series, if you observe any one of them alone, you automatically know the states of all the other ones because they are synchronized. In contrast, synergy is information that can only be learned when you're observing X and Y and Z all simultaneously, and crucially, from no simpler combination of sources. Okay, and so I don't have a pointer up here, but redundancy and synergy are kind of duals of each other. They are related by this very elegant lattice structure. The lattice is not important, but I think it's cool. And I'm happy to talk more about where it comes from and why it has this very particular shape afterwards. So we have sort of the ideas of redundancy and synergy, but what we still need is a summary statistic that we can actually compute given real data. You know, we're still right now in the land of sort of formal idealism, which is where I like to live. And so to compute this, our summary statistic is going to be a measure called the O information. Okay, O for organizational information. And can you guys see my cursor Okay, so the O information is a function that takes in what we will just call a system. This is technically a probability distribution, but we'll just call it a system because the distribution is a model. And if the O information of the system is positive, then this system is dominated by redundancy. Okay, it is more synchronized than it is emergent. In contrast, if the O information is negative, I lost my cursor, then the system is synergy dominated. And all I will say about the formal notation is to say that this measure TC, the total correlation, you can think of it as how much information is there in the whole system. So you take this number, you multiply it by n minus 2 times, and then what you're going to do is you're going to subtract off the total correlation of all of the leave one out marginals. So x to the negative I is the system, every element excluding the ith element. So if this term is greater than this term, then there is some information that is in the whole that persists even after you pull out all of the parts. And then you slap a negative sign on there for sort of weird historical reasons that I won't go into. But this is our summary statistic. And are we all kind of hanging on for dear life here? Okay, so you've made it through the math. And so the question, coming down from the 10,000 foot view to the 5,000 foot view is how does the distribution of emergent higher order synergistic information as indexed by the O information change over the course of infant development? Okay, we have this measure, we have some data, we have this measure, we have the questions, and so now we need some data. And specifically, we needed babies. And we went looking for babies and we found them at Columbia University. And so we had about 300 infants. And they were, this was data collected as part of a project on neonatal intensive care. And so the data was collected from the infants between 33 to 45 weeks post-conception, 124 electrodes that we then source modeled into 57 cortical parcels. And we only used data that was collected during the quiet sleep state, basically just because that made it easier to get long, unartifacted time series, because the O information is fairly data intensive. And then what's really cool is we also had cognitive testing done about 2 years later using something called a Bailey score. And I could not tell you anything about Bailey scores beyond that they measure something like infant IQ or toddler IQ. And so the hope is that we can learn something about how the structure of the information architecture in infancy informs on the future cognitive performance of the children later on in development. Some of the infants were also involved in a clinical trial called the family nurture intervention. And so this was a test to see how enhanced mother-infant interaction could facilitate positive outcomes for babies that were in neonatal intensive care. And so the babies that were in the FNI intervention, they got an extra 6 hours of time with the mother, skin-to-skin contact, calming sessions supervised by a family nurture intervention expert. And all of this was sort of designed to help facilitate autonomic co-regulation in infants that are in a very stressful situation. And so we had 60 babies that were just getting regular NICU care, and then another 74 babies that were getting this extra family nurture intervention. So we're trying to connect the information architecture of the brain with cognitive development, and then the social emotional bonding with the mother as a sort of a triangle. Okay. All right, the last piece of conceptual machinery that we need is something called a simulated annealing algorithm, okay? And the simulated annealing algorithm is an optimization algorithm that is designed to find the best set of cortical brain regions that has the most negative O information, which we call the most synergy. And we did this because we wanted to see how this changed over the course of scales. We didn't just want to look at the whole brain. We wanted to look at, the sets of 3 brain regions, the sets of 4 brain regions, the sets of 5 brain regions. And so you'll see a lot of these plots where you have the optimal O information plotted against different subset sizes. Every dot corresponds to a trial of the simulated annealing algorithm. And what you can see is that there's a very nice sort of U-shaped curve where at around, let's say 10 brain regions, the curve kind of bottoms out. So 10 brain regions is approximately, in this particular trial, is approximately where the most synergistic information can be extracted. That's, what we might call a natural scale with the most synergistically informative degrees of freedom. As you continue to increase the number of brain regions, eventually just redundancy swamps and your signal becomes too compressible to be interesting. So what?
So it's not big. So
Yes, they were source modeled to 57. Yeah, and you'll see a plot. You'll see a plot later. And so, we started just looking at the whole brain O information. So there's no simulated annealing here yet. Every dot is a baby. You can see conceptual age on the x-axis, whole brain O information on the y-axis. And I've shown, 4 frequency bands just for the spirit of completeness, but everything is always the same across all 4 frequency bands. So just pick your favorite one and look at that. And so you can see that the very, very premature babies at like 33 weeks, they are extremely redundancy dominated. This is a very high positive O information, okay? The brain data is highly, highly synchronized. And then as development occurs, going from 33 weeks to 45 weeks, you see this drift from the redundancy dominated condition all the way down to 0. And in the high frequency bands, it actually crosses 0 into a synergy dominated regime. So you're really seeing a transition from 2 very different information architectures, one dominated by compressible copying of information to one dominated by emergent, what you might call sophisticated integrated information. Okay, and I'm not using that in the Giulio Tononi sense. I'm using it in a much more sort of hand-wavy way. Then if we look at the simulated annealing results on the top, you can see trajectories for these U-shaped trajectories for every baby. But it's easier if we just bin them by 2-week intervals. We've plotted the bottom, the nadirs of those curves by with these little dots. And you can see that as the babies get older, that is to say they transition from purple to yellow, the bottoms get deeper, that is to say there is more synergy overall, and they shift to the right. That is to say that the optimally synergistic ensemble, the number of brain regions, increases as well. So you're seeing over the course of development, not just an increase in the raw amount of this stuff, synergy, but you're seeing more and more brain regions getting recruited into participation in these synergistic ensembles. Okay. And one of the things that I want to mention is that, if you go back to this, each one of these dots is a run of the simulated annealing algorithm. There is not one optimally synergistic group of 5 brain regions. There are, in fact, a huge number of equivalently synergistic brain regions. So there's a lot of degeneracy here. And so, there was some paper recently talking about the idea of a synergistic core, its relevance for consciousness. We're not finding that. What we're finding is something more like a synergistic scaffolds, which I'll talk about later, of multiple overlapping sets of brain regions. And so then if we look at how different individual brain regions, how frequently they are recruited into these synergistic, these optimally synergistic ensembles, we've got over columns are our frequency bands, and then over rows are our ages. And you can see that early on in development, where there is less synergy, the synergy that is there is all in the frontal regions, with just a little bit in the occipital regions. And then as development increases, as those synergistic ensembles become larger and more potent, more and more brain regions are recruited until by the end, you've got to 40 to 43 weeks. Every one of these major sort of cortical parcels is included. You've got frontal, central, temporal, and occipital all contributing. So we're seeing a kind of a growth that starts in the frontal cortex, then begins recruiting the occipital cortex, and then finally the entire brain is integrated into this sort of higher order structure. So again, not a synergistic core, but a synergistic scaffold. Now looking at the cognitive performance. So on the X, you've got Bailey scores. On the Y, you have the whole brain infant O information. And so remember that more negative is more synergy. So the negative relationship here says that those babies that were scoring very highly, or not babies, toddlers that were scoring very highly on the Bailey scores at 2 years of age, they had more negative O information at the whole brain level in infancy. And, this is, even if we regress out for age, you still get this effect. And so this is very promising to us because it tells us that this isn't just some kind of numerical shell game where you give me data and I give you arcane statistics back, but this is actually telling us something about how the development of information processing architectures in infancy tracks tracks later cognitive development. And then, so finally, when we look at the family nurture interventions, we can see that the red babies, that is to say the FNI babies, they have a very smooth, almost linear trajectory. Again, this is whole brain O information on the y-axis, time on the X, And the red, the FNI babies, they have this smooth, almost linear transition from redundancy dominated down towards synergy dominated. While the standard care babies, the ones that are just in the NICU boxes, they retain a redundant architecture right up until almost full term age, at which point they very quickly have to catch up. And they reach the same sort of level of O information, but the trajectory of how they get there is very different. And so this is something that we're still trying to figure out what this means, but it suggests to us that mother-infant social interaction is also involved in the emergence of this synergistic scaffolds. And then so in the interest of time, summary, all the stuff I just told you, I want to take just a moment to kind of speculate wildly here. We know from work by one of my collaborators, Andrea Lupi and Emmanuel Stamatakis, that loss of consciousness in adults following anesthesia or traumatic brain injury is associated with a collapse of synergistic structures in adult human brain data. Okay, so again, we already talked about the issues of generalizing from adults to infants. I also want to note that anesthesia and TBI are gross perturbations to the brain. If you run over somebody's head with a truck, everything changes. You lose synergy, but you lose everything else as well. The structural scans are extremely, cortex is thin, ventricles have expanded. So I don't know how much weight to put on those particular results. But I do think it's worth mentioning that the change from redundancy dominated to synergy dominated is gradual and mostly linear, right? So there is not a phase change here. This is not an easing model where you go from one to the other like that. There's no off to on. So maybe instead of a light switch, we're looking at something more, if the link with synergy and consciousness is valid, we would be looking at something more like a dimmer switch, a kind of gradual illumination that occurs as the brain transitions from highly compressible redundancy to highly structured emergent synergy. All of this and more can be read on the preprint, and that's a QR code that will hopefully take you to the bioarchive link. We are in our third round of review. There was one recalcitrant reviewer who I know some of you know. I will not shame him, but he's been very difficult with the best intentions. But hopefully this will be coming out in communications biology very, very shortly. And then so finally, this work was done, started during my PhD, now continuing my postdoc with Dr. Olaf Sporns at Indiana University. Anton Tokarev and Samsa Van Hatlo are from the University of Helsinki. They are the baby experts. And then I, of course, I'm coming to you from the University of Vermont, where I work at the newly christened Vermont Complex Systems Institute. We're now an institute, no longer a center. So that's very exciting. Yeah.
Hi, great talk. So I have a question about your criticism of Limpo-Ziv and then how it relates to O information. So you said you don't like Limpo-Ziv very much because it's a compression measure. And so if you have a maximally random signal, then you have maximal Limpo-Ziv. But then you also describe the O information as a sort of spatial compression, right? Because if you have maximally different signals across space, then that will maximize the O information
So,
right?
Yeah, so we're going to get in very, very briefly get into a little bit of math here.
Sure.
The O information is constructed in such a way that if it is very strongly positive, the system is compressible. If it is very strongly negative, the system is synergistic. And I didn't mention this, and if the O information is 0, all of the parts are independent. So a system that has high entropy but no structure could have high Limpo-Ziv and an O information of 0. I did not mention that, but yes, the synergy is distinct from entropy in that you need a certain amount of entropy to support synergy, but high entropy is not. Entropy is necessary but not sufficient for synergy. You need this extra level of structural organization that comes from all of that.
Cool. And one more question, if you don't mind. So I'd be really interested in trying to replicate this with fetal MEG, but we only have sensory evoked data. Well, technically we have spontaneous data, but it's extremely noisy. Do you have any kind of intuition for how this would behave if you applied it to, in your case, ERPs, rather than just sleep data?
So we used continuous Gaussian estimators, which requires stationary signals. And so if you have an ERP where there is assumed to be some non-stationarity, like a response to an event, I would not do that.
Okay, thanks.
Yeah.
Thanks a lot. That was great. Just a different way of thinking about it that might be fun to talk about is rather than think of a dimmer switch, but something like accruing granularity, looking at different ensembles. A colleague and I in Columbia were looking at persistent homology as a way.
Oh, nice.
And it was a lot of fun. And you can actually look at interaction effects at higher Betty numbers. And so love to speak with you about that because I think it'd be a fun twist on the way you're approaching.
So we should talk because I am just in the process of getting ready to preprint a paper showing that cavities in the topological sense are correlated with synergy in the information theory sense. We're trying to link these two approaches to higher order interactions.
That's exciting.
Yes,
That's
they are very, very much linked.
Great.
Somebody back here.
Thank you. This is very, very exciting stuff. But my question is about, I mean, this is the obvious question is, so what is the link between this informational synergy and consciousness and different senses of consciousness, right? So I mean, can you apply this math to informational processing system and just look at the kind of like the capacity to process information and more synergistically, it's capable of serving certain cognitive tasks or solving certain problems, but where does the consciousness part in a way sort of come in? Is that sort of more the speculative part, or do you see that that's like strongly related to the math behind it?
So I have two answers to that. One is that there is some, again, empirical evidence from Emmanuel Stamatakis' group suggesting that synergy changes with level of consciousness. We have been looking at psychedelics. That's kind of the usually other direction people go. Have found some promising signals, but nothing publishable yet. I would say, right now, the assumption, it's kind of an assumption. I don't want to claim that there is an identity between synergy and consciousness. I'm not doing the Giulio Tononi thing. The thought is that synergy is a general measure of the capacity to support any kind of emergent property. The thought is that consciousness may be one of those, or whatever particular configuration is necessary to maintain consciousness should be some kind of emergent property. So that's as far as I'm willing to go. Call Andrea Lupi; he'll be more philosophical about it.
So it's really interesting to do this kind of work where you try to come up with a single number that captures something really important about high level structure, basically, that makes a difference to thought and consciousness and so on. But presumably there are going to be many, many more kinds of high level structure than you can capture using a single real number like this. So in some sense, you're collapsing many, many, many different on the surface, highly relevant kinds of structure onto a single dimension in order to do this kind of work, which doesn't mean it's not interesting, but I think it's got to be just the very beginning of this kind of mathematization of this sort of loose notion of structure that we all presume is doing the work of making people intelligent and conscious and all the rest.
Yeah, so I think that's going to be true of any attempt to make a scalar measure. Like a scalar is it's one dimensional. You're always going to lose something. I also want to say, sort of in defense of our fields, this a lot of this started back in 2010. This was Williams and Beer. The field itself is quite new, and we've spent about it's 2025 now. We spent probably the first 10 years as a field since Williams and Beer, sort of trying to work out all the formal details. So it's a very, very nascent field, and so there's a lot of now these deeper questions about how does this stuff relate to biology or cognition or politics, whatever you want. Now we're just now getting to the point where we can start to ask those questions because we were sort of getting our own house in order for most of the last 15 years.
Thank you. This period of time seems to correlate with increasing connectivity in the brain. Is that fair?
Yes.
And I understand these were more or less sleeping infants.
Yes.
What happens when they have a simple task?
Oh, I have no idea. We did look at active sleep as well. There's in the supplements for the paper, there's all the active sleep results, and the effects are for the most part the same. So at least I can say that whether you're, this is not unique to quiet sleep, but these babies are no longer babies, they're grown, they're long since left the NICU. So we're limited by the data that we have.
But you would think if the infant is doing a task like listening or looking or whatever, eating, you would have more focus and less synergy because you would be recruiting certain parts of the brain and not others.
So I'm about to start a project with Hugh Garavan up at UVM looking at synergy in adolescent brain data. So not infants, assuming that we all still have jobs in a year, get back to me and I will hopefully have more of an answer for you because I don't know right now. I think my prediction actually would be increased synergy during task, and the reason is a little bit sort of mathematical. But if you have a system that contains a causal collider, something where an element needs to decide its state as a function of multiple independent inputs, you will see synergy there. That's kind of built into the mathematics of how a collider works. And so if you are doing a task where you have to integrate multiple streams of sensory information, that's in some very hand-wavy sense, a causal collider in the cognitive configuration space. And so I would expect to see synergy there. But again, I'm more on the math side than the brain side.
Sure.
So
one question.
I was wondering if you could briefly, wildly speculate on particular theories of consciousness, because the synergy in the occipital lobes was a little surprising to me, and it made me think of a couple of options, like some of Giulio's speculations about the phenomenology of space in V1, or maybe even recurrent processing theories, or or is there even a relationship between the synergy in the frontal lobes and the occipital lobes in some sort of higher order thought sense? If you could speculate wildly.
Speculate wildly. I can always do that. So the easiest one to speculate on is IIT, because a lot of this sort of descends from Giulio's original ideas about trying to quantify some notion of greater than the sum of their partsness. I think Giulio comes on way too strong, and I'm not making that kind of identity claim. I also think that IIT has become quite baroque in how you compute it. I think synergy is much more elegant. As for other things like higher order thought, maybe it would be a natural hypothesis that higher order thought might be represented in the hardware through higher order synergistic dependencies between neurons. I have no evidence to back that up, but it seems plausible. Global workspace has always been kind of a question for me because I don't. Andrea Lupi has a results about that actually. Now that I think about it, and I don't remember what it was. We just count how many times each one appears in a bag in one of the optimal bags. We don't look at the interaction between the two different lobes.
Thank you very much, Susan. So the title of my talk is Infant Consciousness: How, Where, Whether, When, and What. And so we'll say a little bit about those 5 questions. I'm not actually giving any argument. It's more, the idea of the talk is more giving a framework of how we should study infant consciousness. And I will, instead of giving my own answers to those questions, I will present the survey and your responses to those questions. So this is the plan of the talk. Okay, so those 5 questions. So the how question is a methodological question. How can we study consciousness in infants? What are the methods? The second question is the where question. So where in the brain is infant consciousness located? What is the neural basis for the infant consciousness? The other question is the existence, whether newborns, infants are conscious. We could ask this question for other creatures, but in this conference, we're addressing the main phase for us, newborn infants. The one question is that we can't disambiguate the rather question if newborns are conscious or not from when consciousness emerges in development. So the when question tries to capture this, the emergence of consciousness in human development. You might think that maybe the capacity is in place earlier than when the capacity is deployed, for instance. And the final question is the what question. What is infant consciousness like? So what kind of phenomenology they might have. I don't think those questions are the only questions. There are further questions, but I think they're the main questions. You can also ask about the function of consciousness, the why question, and you can also ask the question, the which question, okay? And I think those the survey tried to go further than those 5 questions, but I think they are the main questions, and I will say a little bit more about the questions. So I'm not the first one to propose this approach, this framework for consciousness. For instance, Ned Block, in his entry for the Stanford Encyclopedia in Philosophy, his entry is based in some variations of those questions. What are the features of consciousness? How consciousness exists? And why does consciousness exist? Joel and Chalmers, in a recent paper, Markers of Consciousness, they have some versions of those questions. Which markers constitute a cluster? When does consciousness begin? Hugo Lagergrants, that were mentioned in many talks during the conference, one was a pioneer in infant consciousness. He also has a classic paper where he has a variation of those questions. So it's just an attempt to systematize what are the relevant questions and how we could reply to those questions. We can also see the program of the conference and try to map talks on those questions. So for instance, the first keynote talk by Lorena Nachi and the first symposium, Neural Correlates of Consciousness or Infant Consciousness, I think they're trying to answer the rare questions, where are the neural bases for infant consciousness. I think Ghislaine talk maybe was whether newborn infants have the inner drive, and so maybe rather questions about consciousness. I think the last symposium yesterday on fetal consciousness, we're trying to answer the when question. So when consciousness emerged in human development. The first talk of today by Tim Bain was about the methodology, so how we can study consciousness and the markers, potential markers for consciousness. And the infant caller session, I thought, was trying to answer what kind of phenomenology, if caller, the experience of caller would be one of those type of phenomenology or not, so sensorial phenomenology regarding caller perception. And this last symposium is a mixed bag of what and rare questions probably. So it's a less focused symposium, but I think it covers some of those questions. Okay, so now a little bit of what I think each of those questions try to capture. So how can we study consciousness in infants? So this is a summary of most of you said a little bit about this. So we cannot directly observe consciousness in others. In other humans, we rely on verbal reports. Infants, we cannot use verbal reports or come under following. We cannot rely on past memories, although I think Nick is trying to convince us that there are more there than we thought before. So maybe memories might be a good way to start looking for. But what evidence can we use? So we have, to answer the how question, we have those 3 potential methodological approach: neural markers, behavior markers, or theories of consciousness. There is a discussion what of those methods we should favor. I think Tim Bain gave an excellent talk, explained the difference between what would be a marker first approach or a theory first approach. We can also suggest a hybrid approach, maybe Jonathan Bert's approach is an attempt to have a hybrid approach. You don't start with the theory, we start with a marker, a behavioral neural marker, but the theory will inform whether those markers should be more relevant. So there is some role for the theories in a hybrid approach. Okay, so the rare question, so where is the neural basis of infant consciousness? We can divide these questions in three potential responses. Either the neural basis is a sensory basis, or is a frontal cortex basis, or a subcortical system basis. Okay. Whether newborn infants are conscious is a straightforward question. Whether you think or not they are conscious based probably in the methodology you think is more appealing, or whether the markers, the behavioral or neural markers are a type of evidence that would settle the question. So there is yes or no for this question. And you can have this similar question for self-consciousness. So when infants are self-conscious. So when does consciousness emerge in human development? So recently, Tim Bain, Lorena Nati, Joe Froelich, and others have been proposed to divide this question in two big areas. One would be early onset of consciousness and a late onset of consciousness. I propose a more fine-grained approach to this question, most because I think some of the early and the late approach, they end up catalyzing different views in the field. So one way to reply to the one question is think about those 5 answers. Either you endorse an early prenatal view, so consciousness emerges in fetus before 24 weeks of pregnancy, or a late prenatal view, consciousness typically emerges in fetus after 24 or more weeks of pregnancy. The natal views, those that think that birth actually plays an important role, so consciousness emerges at birth. Early postnatal view of consciousness, so consciousness typically emerges in the first 6 months after birth, or a late postnatal view of consciousness. A consciousness would emerge 7 months after birth. Okay. Okay. Finally, the what question. So what type of phenomenology newborn infants have? Sensory experience, just perceptual consciousness, or affective experience, pain and pleasure. Cognitive experience is experience of thinking, or agentive experience of acting. This probably varies with what type of neural basis you think is the neural base of newborn infant consciousness. Okay, so as I said in the beginning, instead of giving my own answers to those questions, I will turn to the survey and see what this community think about those questions. This is a joint work with David Chalmers. So we don't think that the survey gives us the truth of those those questions. So it's not, but I think this is an important community. There are lots of experts in this community. So whatever the response that is more endorsed by this community, I think it might be a response, at least you have to take seriously as an important response. Okay, so now I will turn to the survey. If you didn't take the survey, you can still take the survey, not for the purpose of this talk, but we'll continue collecting responses until we finalize a number of responses we think that has some sort of statistical power for the survey. Okay, so demographics. So this is a conference organized by 4 philosophers, Matthew, Ned, Dave, and I. So 53% of the participants in the survey. So the majority is, so this is a survey that the majority was philosophers. So might make an important differences in the way the questions are answered. But you also have important representation of psychologists and neuroscientists and other academic fields. Okay, so 35% or 50% of the answers come from experts that work on infants, but the majority, if you don't not work infants, there are a huge part of the demographic that work on consciousness. So it's experts on consciousness that actually are replying to this question. Okay, so the whether question, newborn human babies typically conscious, 8% said yes, just 5% says no, but surprisingly, there is still agnostic. So almost 14% of people are still agnostic what is going on in newborn human babies. The when question, I also thought this is a relevant response. So I don't think some years ago the late prenatal would be the phase that people thought that consciousness would emerge. So 9% think that early prenatal, 47% late prenatal, at birth 11%, early postnatal 12%, but we still have 5% of response that consciousness might emerge late than 6 months or after 6 months. Okay, so but I think there is a shift of at least some endorsement of a sort of late prenatal emergence of consciousness. Okay, the one question for self-consciousness, self-awareness. So the majority think that, I think there are less disagreement on this, just few would think that self-consciousness would emerge in early prenatal stages. There is just 8% that think the late prenatal onset would have an onset at 8% of people think that self-consciousness would emerge at late prenatal phase. At birth, 3%, and early postnatal, 20%. But the majority think that is after 6 months that self-consciousness might emerge. Okay, the where question. So, although there is a lot of answers that didn't compromise with any of those neural bases. So 44% of response are agnostic, where is the neural basis of infant consciousness, but the majority think that is in the sensory cortex or subcortical systems. But so just 20%, less than 20% think that the frontal cortex would be the neural basis of infant consciousness. So now the what question. So almost 90%, 87% thinks that infants have affective experience of pain. But there is still a 10% of agnostic whether they have any phenomenology at all. And 79% thinks that infants have sensory experience, and the minority think that they have cognitive experience of thinking, and agentive experience of acting. Okay, you can, the question about the markers, the methodological question. And although we discuss a lot of the neural markers in this conference, the behavioral markers are still the the most popular markers for consciousness in infants. So 62% favor the behavior markers, 55% neuromarkers, and just few thinks that theories would be relevant as a guide to infant consciousness. So theories is very unpopular those days. Maybe the marker approach is actually winning. Okay, the which question in the sense, so one question we can ask is the distribution of consciousness in different systems. We plan later to compare those responses to the newborn infant's response. So of course, I'm happy that 100% think that adult humans are conscious. So it seems you don't have a doubt on this. Yeah. So less of agnostic. What? Okay, cats, 93%. Fish, surprisingly, is getting higher and higher, 72%. Flies, 41%. Plants, we still have plants. Some people in this community that think plants are conscious. And future systems is getting higher and higher. You got 37% of potential consciousness. Okay, I'm seeing that we have the newborns. Yeah, we just have here. Oh, yeah, 8%. Okay, yeah. So babies is less than cats. No, I hope not. This is a serious survey. Don't joke. Yeah. Yeah, no, I totally agree. Yeah. I think one interesting correlation is the marker you favor and which systems you think are conscious. Are there any correlation between the markers and? Yeah, yeah. Okay, so those which of those theories do you find the most promising? I know Ned will be very happy. Biological theories are winning. So he is favoring his view. But we still have important credence on global network space theories and integrated information theory and higher order theory. And okay, there is another one here. Oh, yeah, the recurrent processing theory is not as popular as we could expect. Yeah, okay, before the question. Okay. Yeah, no, I just have one slide and just to we're having a special issue on infant consciousness. It's edited by me and Sasha Fink. Will be a special issue of the Philosophy and the Mayan Science Journal, and submissions are very welcome. We will launch the call for papers soon. Okay, thank you.
Yeah, thank you so much, Claudia. This is really wonderful. I have one comment and one question. So the comment is we really need to be super careful about the demographics and the population, because I have this hunch that philosophers are now representative of the general population. And actually I have a dream of mine, just like the next workshop that I'm going to do. I'm basically going to do interdisciplinary audience and collect neural and physiological data on the people when they do the workshop, because researchers research stuff, but nobody researches. And I think it's super interesting because they make big claims, and have this dream of like me putting philosophers in the scan to see what is different from the general population, because I'm pretty sure I'm going to see many differences. So that's the comment. But I think my question is about the list of the important questions that you have. I think there is one which is super important. It's not there, which is how consciousness emerge. It's not how we measure, and not when, where, how, but how actually does it happen? So now the how question for me was not how we measure, because yeah, of course, just like this is things that we can measure or not. But then I think the really important question is like, how exactly is it happening? And then I see why the biological theories they are so popular, because then the question would be was like how you get self-organization in inert system versus living system, and then to which degree does biological self-organization leads to some sort of like, you know, high order, forget what I said, not high order, sophisticated way of like experiencing. So for me, that's the core question that needs to be added to that list, and it's not there because the rest is kind of like easy. if you get, I mean, obviously, you know that I disagree with this idea that we should somehow find it in the brain. I don't think we're going to find it in the brain. I think this is loss of energy. I think we need to look at physiological interactions between, you know, different systems in the body, endocrinology, immune system, physiology, and neural. Yeah, so what's your comment on that?
Yeah, thank you. Maybe this. So this is those questions I'm developing in a paper will be out soon. The last session of the paper is like, what are the further developments? And maybe I captured the question you're trying to press me on in the why question. So what's the function of consciousness? So why we are conscious? Maybe this could be a way to capture what you have in mind. I don't have like a good answer for this question, but Ned and Dave are organizing a conference on the function of consciousness or workshop, maybe 12 this year. So I invite you all to come. But I think it's a good question. So why we, maybe how consciousness evolves could be captured. I'm calling this the why question, the function of consciousness. Okay, thank you. Yeah, you opposed to what we have in the survey. Why related to the theories? Okay. Yeah.
For the which question, I was wondering whether you were looking for confident answers or just kind of answers we were inclined to give. Because I remember when I was filling out that question, I wasn't sure whether to put down or whether to say yes to things where I think it's more likely to be the case that members are conscious, or that some members are conscious, but where I'm not sure. So, like I think it's more probable than not that some fish are conscious, but I didn't put down that I didn't put down yes for fish because I don't feel confident about that answer. So I guess I was just wondering how confident of an answer were you looking for it to constitute?
Yes, good. So this question, the which question, was not the question we developed, was a question from few papers survey. So maybe I defer to Dave because we just copy-paste the question.
Actually, in the instructions for the survey, I don't know if everybody read them. We asked people. Sorry, I know that who reads the instructions for a survey anyway, but we did suggest that people respond with the view they lean toward, rather than views that they have, rather than requiring acceptance or certainty. On the Phil Papers survey, we gave the choice: accept or lean toward. But there was also a part where we defined consciousness, by the way, in terms of subjective experience and what it's like. But I think the instructions were to respond with a view, even if you lean towards that view without accepting it.
Okay.
Hi, Claudia. So I have a question about your passion for the research. So I've actually never considered infant consciousness before. I heard about your research, and then I heard about this conference. I thought, I've got to go. We hear about, are animals conscious or machines conscious? But I think the question of like, when infants become conscious or whether or not infants are conscious really had never occurred to me. And so it's really exciting to hear all of this research from all these different fields of people working on this. So I'm curious how you came to being interested in this topic, since you seem to be the, you're one of the main organizers and it's your research. So I just wanted to hear like where the passion, like, I don't know, why infants, right? Like out of all the consciousness. Yeah.
Yeah, I can respond from my own perspective. It probably is a kind of a personal story and very boring. Maybe there are other experts here. I don't know, Ghislaine has been studying infants her entire life, so probably has a more better approach for why this, because in the question of why this topic is relevant. So I started my academic career interested actually in introspection and self-consciousness. And for understand self-consciousness, I realized that I had to understand what's happening in the early stages. And there wasn't too much literature on the topic. So I was searching for a philosophical guidance for infant consciousness in early stages. And there was much more hypotheses and questions than actually development of the topic. So I wrote my thesis on 2017 on development of consciousness. And in writing the thesis, I become much more interested in this topic and the markers and the theories. And this become, I don't know, with the advent, the technological advance that you can study better the brains of infants and those brilliant neuroscientists we have here that actually went there to test what's going on in their brains, I think make the field much more interesting and fascinating to just respond to those questions. And this is, yeah.